सदरन ब्लॉट हाइब्रिडाइजेशन में अभी तक हम बात कर चुके हैं जेल इलेक्ट्रोफोरेसिस की, और जो डीएनए सैंपल्स हैं उनको नायलॉन मेम्ब्रेन या किसी भी सॉलिड सपोर्ट पे ट्रांसफ़र करने की, ताकि हम इन डीएनए मॉलिक्यूल्स को इमोबिलाइज कर सकें। अब इमोबिलाइज करने के बाद जो नेक्स्ट स्टेप है दैट इज़ द हाइब्रिडाइजेशन प्रोसेस इटसेल्फ, so that we can detect the DNA of interest or the segment of the DNA which we are looking for. For that purpose, we have to use a probe which is radiolabeled. अगर probe को define करें, this is a sequence of nucleotides that is radiolabeled and it emits the radiations. It is a complementary sequence to that particular DNA fragment which we want to identify. And as you know that the complementary base pairing will be possible if we have probe on one side and DNA on the other side, the single-stranded or denatured DNA, and wherever the probe is going to bind from that particular position, we are going to get some radiations coming which will be recorded on X-ray film. इस सारे प्रोसेस को जब देखा जाता है कि हमारे पास probe hybridization के कौन कौन से factors हैं which are going to affect the hybridization process or the results. The first one is the proportion of immobilized DNA and the probe. हमें पता होना चाहिए कि हमारे पास जो DNA हमने immobilize किया है उसकी quantity क्या है, और accordingly हमने जो probe की quantity use करनी है वो कितनी use करनी है. अगर इसमें proportion नहीं होगा the result is not going to be very good. उसके बाद size of the probe. A probe की length can be from 200 nucleotide up to several hundred nucleotides. It can be 100 nucleotide as well. But in that case, the length or the size of the probe is going to give you false results as well if you are going to use very small sized probe. So the probe should be quite much long as possible for hybridization. And जब हम probe prepare करते हैं उसमें हम radiolabeled nucleotide use करते हैं, और at the end of the probe synthesis जो हम आगे चल के बात करेंगे, the specific activity is measured in terms of counts per minute CPM. एक अच्छा probe जिसको हम hybridization के लिए use कर सकते हैं, it should have a specific activity around more than 1 billion CPM per microgram. Then amount of genomic DNA that is transferred from gel to the membrane, that is going to determine the best results for hybridization. Next, we have to think about how much sensitive is this probing method. कि हम एक रेडियोलेबल प्रोब यूज कर रहे हैं, ये कितना मिनिमम पॉसिबल डीएनए जो है उसको डिटेक्ट कर सकता है. हाउ सेंसिटिव इज दैट? इफ वी हैव बेस्ट कंडीशंस ऑप्टिमाइज्ड, दैट मींस टेम्परेचर, आवर हाइब्रिडाइजेशन बफर, द लेंथ ऑफ द प्रोब, then it can detect less than 0.1 picogram of DNA which is immobilized on the membrane. Keep in mind this limit or this sensitivity will be observed if the probe is having its specific activity more than 1 billion CPM per microgram. Hybridization का process complete होने के बाद we have to expose the X-ray film to the nylon membrane which has been hybridized with the probe. अब इस X-ray film को कितनी देर के लिए expose करना होता है to get some detectable signal? It depends upon the size of the fragment. अगर हम 1000 base pair के DNA को expose कर रहे हैं, the X-ray film should be exposed to the probe hybridized sample overnight at minus 70 degrees centigrade. In that case, we should assume that we have used 10 microgram of the genomic DNA which is transferred to the membrane and is hybridized to the probe which is around 1000 nucleotides long. Besides these radiolabeled probes, we now have some non-radiolabeled methods available. These non-radiolabeled method have some plus points or some advantages over the radio-labeled method. For example, they have or they need less exposure time. They have longer storage times, but at the same time, they have some drawbacks. And these drawbacks discourage us to use this non-radio-labeled method. For example, this method is not going to detect a single copy sequence through the Southern blotting. Backgrounds, they are quite often very high, and the signal is not exactly proportional to the amount of DNA which is immobilized on the membrane.